Hello, judges. My name is Libei Zhu from Basis International School, Hangzhou, and my project title is quite long. The overexpression of RAC2 in the Alzheimer's disease brain increased microglial phagocytosis. So first, I will briefly introduce the Alzheimer's disease. It is a degenerative neurological disease characterized by the memory impairment and the loss of neurons. Amyloid beta protein accumulation is considered as the main reason. In response to the beta accumulation, microglia become activated and produced pro-inflammatory cytokines, which will worsen the neurodegenerations and contribute to the AD progressions. However, there's still no cure due to the limited understandings of its pathogenesis. Microglia are resident cells in the brain and could remove dead cells and cells debris to maintain the brain homeostasis. Overactivated microglia are a well-characterized histological indicator from the AD brains. Further research is necessary to fully understand the mechanisms by which the microglia contribute to AD and develop new strategies for the AD treatments. Now we're moving on to our research questions. The A-beta clearance in the microglia are mediated by many pathways. My research aims to explore how the risk genes participate in the pathogenesis of AD. My hypothesis is that addition or the absence of some risk genes efficiently affect the microglia functions, especially on cleaning of A-beta debris. So what are the processes and the plan for the experiment? We first collect the published RNA sequence samples from both the AD patients and the control, which are the normal people, and the data were clustered by the cell type to compare which functions of these genes regulate microglia. We performed gene ontology analysis using the SR plot. RAC2 was selected, which is involved in the microglia phagocytosis. Mouse microglia cell line, BB2 cells, and A-beta burden microglia AD cell model were used for the following in the experiment. When RAC2 was overexpressed, the cellular morphology, activated state, and the phagocytosis ability were observed. At last, the possible molecular mechanisms were explored and discussed. So I'll use the images to show my results. This is the data for the cell clustering. We will only focus on the gene expression for the microglia. And the volcano map over here shows the gene expressions differences between the normal and the AD patients in the microglia. And gene ontology results show that most of the differentially expressed genes clustered in the cell pathway are associated with phagocytosis and the cell membrane activity. Thus, the genes may affect the immune functions of microglia, especially the phagocytosis function. Among that, RAC2 was reported to regulate the immune response. So its function in microglia was selected to explore the following cell experiments. Since RAC2 upregulated in the AD brain, we will construct the RAC2 overexpression plasmids. After RAC2 overexpressing, BB2 cells shows activated response with more protrusions. In addition, RAC2 overexpression induced M2 activations of microglia and downregulating M1 activation. Real-time PCR data show that the typical cytokines indicating the M2 type activations were increased. At last, A-beta burden BB2 cells were clustered in the AD cell model, and ELISA analysis shows that the RAC2 overexpression did make the microglia clear the A-beta protein faster. So lastly, it's the discussions and the future plans. Previous research had proved that the upregulating expressions of RAC2 is closely related to the microglia inflammation response and AD, consistent with our results. Here, we discovered the upregulating of RAC2 in the microglia could promote the A-beta clearance. Studying the link between the microglial phagocytosis and the A-beta clearance could provide more insights for the AD treatment. And more importantly, upregulating of RAC2 might be a potential gene-based therapy for the A-beta clearance. The recent success of gene therapies in other neurodegenerative disease highlight the way forward. So here is the references for my project and the researchers, and thank you all for your attention.